So today's exploration, I see some of you asking in the chat. Jeremy Moore will be, he's a graduate student in microbiology and will be talking about bacteria and how they sense their environment. So I will go ahead and hand it over to Jeremy.
Okay, hi, I'm Jeremy. Let's see, this is more people than I've ever been to talk to before, so this is very exciting. I'm very nervous. So anyway, let's just get started. First, I'd like to start with a bit about me. So originally, my family is from Canada, not America, but I grew up in Michigan. I grew up in this sort of rural town called Midland in the Middle. And we have two things. We have two things and only two things of interest. The first is the Santa house, which is a house dedicated to Christmas all year round, where Santas from all over the world come to become the best Santas they can be. And we also have a tridge, which is like a bridge, but three-way, and it's much smaller than it looks in this picture. It is for walking only. There are no cars. It is a small town. So after sort of growing up, going to high school, I went to college in Ohio, and I went to college in a literal village in the middle of Ohio called Gambier. There was a village market, and there was a village coffee shop, and that was it. So we had to drive 20 minutes out of town to a city to get groceries. It was awesome. But while I was in college, I joined a laboratory where I started washing dishes. And after I washed dishes for long enough, they decided to let me play around with some bacteria. And I've been playing around with bacteria ever since. And playing around with bacteria took me here to Connecticut, where I've joined a lab. I work with this tall Swiss dude named Kiri. And this is my sort of direct boss, Keita Kamino. And together we study how bacteria make decisions. And while we're going to talk a lot about decision making in a little bit, I do have one more section about sort of me personally, and that's some hobbies. My first hobby is playing old school arcade fighting games. So I've got like an arcade stick right next to me, actually, but I won't show you because it's super embarrassing. But I just love old school arcade games. So everything being shut down is super unfortunate because there are no arcades. And I'm also super into synthesizers, like making funky space noises. And like the nerdiest way to make music of all time is with what's called a modular synthesizer. Mine is actually right up there, where you have to like wire together electronics together in a way that makes cool, funky noises. So if anyone has any questions about space music, I want to live on a spaceship. So anyway, that's enough about me. Let's move on to the actual science. So some goals for today. So the way I've structured this talk, things are going to get a little bit more complicated as we move on. Things are going to start fairly simple, kind of intuitive. And then as we move on, things will get more and more complicated. But today, all I want you to come away with is to appreciate that bacteria are awesome, and all of you should be studying bacteria for the rest of your life, like I am, because they're super cool, and there's so much to learn about them, and they do so many awesome things that we don't know about. And also to appreciate that you and bacteria are pretty similar in a lot of ways, and that isn't supposed to be a diss, that is supposed to be showing you just how amazing bacteria are, that they solve a lot of problems that you and I come across every day. And before talking about this problem solving, I first want to convince you about part one, which is that bacteria are cool. And so this is yogurt. Everybody loves yogurt. I actually really hated yogurt until this year. Now I like yogurt. And this is sourdough bread. I also really hated sourdough bread until this year when my girlfriend started baking it. And now I think it's delicious. And yogurt and sourdough bread are only possible because of bacteria. This is a bacteria called lactobacillus, and what it does is that it eats the sugar that's in milk and it eats the sugar that's in flour, and it poops out this really sour stuff called lactic acid. And so like the bacteria poop is basically what gives yogurt its tasty sour flavor, and it's also what gives sourdough the sour in its name. There's some other cool bacteria too. There's like billions of them. In your stomach and on your skin, there's this bacteria we're going to talk a lot about today called E. coli. And these are bacteria that look sort of similar to this, except they have a lot of little tails. And these tails are called flagella. So try and keep that in mind, that E. coli is a bacteria that has tails that allow it to swim, and these tails are called flagella. But just so I am being ethical and not convincing you to go make friends with all the bacteria in your world, there are some that you should try and stay away from. Thankfully, because of public health and you know, medicine and soap and stuff, they aren't so common anymore. But some of these are really bad, like cholera. Cholera caused a lot of crazy epidemics in South America and Western Europe hundreds of years ago. And then Legionella. I actually don't know anything about Legionella, but I went online and I looked for a picture of really gross bacteria, and this was like the first hit. So I picked it up. They look pretty gross, which means they're probably bad. But this one I do really like, and this is Lyme disease. So growing up in rural Michigan, when you go into the woods, because all you do in Michigan is play in the woods, when you come home, your parents will like scan you for ticks, or like eventually you'll scan yourself for ticks to make sure you don't get a tick. And you don't want ticks to bite you because of Lyme disease. And so I grew up thinking that Lyme disease was because of ticks, but it turns out it's actually these sort of spirally bacteria. And because scientists are notoriously bad at coming up with names for things, these are called spirochetes just because they're spirally. I'm sure there's some Greek reason for that. But anyway, these are super cool, and they lead into what we're going to talk about, because these bacteria make a lot of interesting decisions. They live in tick spit, but they don't grow in the ticks. They grow sort of out in the wild. And so what they do is they'll be sort of out in the wild, and then they'll smell tick spit and make a decision to swim towards the tick spit. And so understanding how cells make this kind of decision, how do I swim towards or away from something, is really where my research, what my research is interested in. And so I know it's a bit early on, but we're going to have a breakout activity to get us in the mindset of thinking about how bacteria make decisions like this. And to do it, we're going to think not about bacteria at all. We're going to think about pie. So I want you to imagine that you're at your house in your living room, and somewhere in your living room, I have baked and hidden a delicious blueberry pie, or whatever pie flavor is your favorite. And what I want you to do first in this activity is to write down a very specific list of instructions that'll allow you to find the pie without using your eyes. So we just talked about, carrots giving you great eyesight. Imagine that you've never had a carrot in your entire life, and so your eyesight is terrible. To find food, you have to smell. And so using only your sense of smell, I want you to come up with a very sort of specific list of instructions that you can repeat over and over again to find the pie. And a bad example I came up with is to just walk forward, turn your head and sniff, and then spin in a circle and walk backwards. Like, that is not going to get you anywhere near the pie, but that's an example of a list of instructions, the types of actions you're allowed to do. And if you guys come up with a list of instructions like this, the second level to this activity is that you're no longer in your living room. You're in your grandma's living room, which for some reason looks exactly the same, except that she's filled her entire house with candles that smell exactly like your favorite pie flavor. Does the strategy you came up with in part one also work for finding the pie in grandma's house? So something that we talked about in our group that I think is important is that to find a pie in a room like this, you need to know that in some directions, the pie smells stronger than others. And so sort of instinctively as humans, we'll turn our heads whenever we smell something strongly. And so the idea once you get out of this is that smells can be strong and weak, and that as you're closer to the pie, the smells get stronger. And so I'm going to move a couple slides ahead. And this is really the idea of gradients. So gradients is a word that just means that things get larger or smaller as you move in space. So if I'm on the right side of the room, then there's not a lot of smell. And if I'm on the left side of the room, that there's a lot of smell. And this is a gradient. It means that on one side, there's a lot of pie smell, and on one side, there's no smell at all. And so a strategy you could use is to turn your head towards where the smell is strongest and move in that direction, and then periodically re-smell and readjust depending on where the smell is strongest. And if people got to part two, that's okay. And if people didn't, that's also fine. But the basic idea here is that this problem is more difficult if you filled your house with candles. And the idea is that the candles are a background smell, which means a smell that's everywhere in the room that's sort of diffuse. It doesn't have a gradient, but it's always there. And it makes it a little bit more difficult to detect the gradient coming off of the pie. And the reason for this is that if you can smell something everywhere, it's a little bit more difficult to tell the difference between a lot of smell and a little bit of smell. And what we're going to find later in the talk today is that telling the difference between big and small smells with and without backgrounds is exactly the problem bacteria need to solve in order to find food in the wild. So before we talk about bacteria, I just have this cool video, which I think contains a lot of these ideas. This is a really famous video. I've pretty much just included all of my favorite videos in this talk. So what this is a immune cell. So everyone's talking about getting this COVID vaccine. The reason you want vaccines is to build a lot of immunity by making a lot of these little soldiers that are going to go and eat bacteria if they get inside of your body. And so what this immune cell is doing in this video is it's chasing a bacteria, which are these little sort of dumbbell looking guys that show up black in this video. And it's chasing it, which means that it can smell the bacteria, like it knows where it is, but it also knows to ignore this bacteria. It's decided to chase this bacteria and only this one and ignore everything else. But that's not the only decision going on in this video. There's also the bacteria, which can tell the difference between whatever these sort of large blobs are and this immune cell that's chasing it. It needs to smell where the immune cell is and run in the opposite direction. But it also can't run in the opposite direction of any of these other cells. So it needs to know what it's trying to smell and then figure out which direction is best to swim in. And so this system is a little bit complex. There's a lot of things going on, but the core idea of making decisions about where to swim towards or away from, I worked at understanding using this cell I mentioned earlier on, E. coli. This is another really famous video. It's grainy because it was captured on a really old microscope. But what it is just a video of E. coli swimming around in a suit. And I think this shows the types of motions that these E. coli can take. If you watch, some of them are swimming pretty straight. They swim in a really straight line. But if you look very closely in this part of the video, did Jeremy say soup? I did say soup. Actually, turns out the stuff you grow bacteria in is basically soup. So the original stuff people grew bacteria in way back in the 1800s was literally beef broth. So like beef broth you would get in like the store, they would like grow bacteria. And so basically what we grow them in nowadays is just artificial beef broth. I actually don't know how true that is, but that is the legend that I have heard. So, but anyway, so these cells, they're swimming in straight lines. And if you look very closely over here, they'll splay out and sort of spin randomly sometimes. And this random spinning is called a tumble. So when the cells are swimming straight, we call that a run. And they're running because for some reason, when the tails spin counterclockwise, it makes the cells go straight. But if the tails for some reason spin the other way, clockwise, it makes the cells spin around in a random circle. And then eventually it goes back to running. And this sort of spinning around randomly is called a tumble. And if you watch these bacteria swim, they alternate between running and tumbling somewhat random. So what this is if I watch this video very closely and track the position of every bacteria in time, I can make a graph like this. And so this is how a single cell swims. It moves straight, and then here it tumbles. Oh, whoops. And then here it moves off straight again in a new direction. And all of these cells are doing this all the time. It's called running and tumbling. And it seems a little bit weird that this is how they would choose to move. Sort of like we talked about in the example, when we're trying to find food, we determine where the best direction to go is, and then we go in that direction. It's also sort of like driving. Like if you're driving a car and you want to go to Walgreens, you're driving on the road, you see a Walgreens, and then you turn into the parking lot. But what these bacteria are doing is basically seeing a Walgreens and then spinning out and then picking a random direction to go in. It doesn't seem like this would be a strategy at all for actually getting towards food in the environment. But it turns out that cells have to do this. They have to do this if they want to find food. And the reason they have to do it is because they're very tiny. Now, that seems completely disconnected to finding food, but it turns out being very tiny makes the world look different to you than it looks to you or me. So if you and I had special goggles that let us see where food is in the environment, we could take a look at, some soup that for some magical reason had set up a gradient and see that on one side of the soup, there's a lot of food. And on the other side, there's barely any food for some reason. Just bear with me here. But if you're really small like a bacteria, right in front of the bacteria, the amount of food looks the same as the amount of food that's right behind you. Now, this is a little bit counterintuitive, but bear with me. And the cells run and tumble to explore their environment, to essentially like sniff in a wider range around themselves than they can if they stayed in place. So while you and I can tilt our head and then smell different things, if the bacteria spins in a circle, things look the same. So it explores a little bit out from where it started, takes a sniff, and explores back a bit, takes a sniff. And if the food is increasing, it decides to keep going. And if the food is decreasing, it decides to stop, spin around, and try a new direction. So this running and tumbling is really a way to solve the issue of cells being really small and unable to tell the difference between more food and less food in front and back of them. And so I hope you can see that this is a strategy that eventually solves the pie problem, just less efficiently than you or I could. And to see that, imagine the bacteria starts over here, and it goes over here, and as it's swimming, the amount of pie seems to be increasing, so it keeps swimming. But eventually, it'll just randomly switch directions. But the amount of food now is decreasing, so it spins around until it finds the direction where food is increasing. And eventually, by repeating this over and over and over again, bacteria are able to find food. So I think I'll pause here for questions. There's a little bit more to go through. But if people have questions, you can either unmute yourself or maybe some people can read questions from the chat.
I can read questions. The students are very curious. One, why do bacteria tumble? And there's a bunch of questions about tumbling and tail turning. So the first is, why do bacteria tumble?
So I'm going to assume that means like, why does it tumble when it goes clockwise versus counterclockwise? So that is actually kind of mysterious. That's a very interesting question. And if anyone wants to figure out the answer to that question, they should study bacteria. But to our best knowledge, it has to do with the way that the tail screws. So if anyone's ever played with like a screwdriver, you know that the screws only go in one way. And that's called handedness. So if you have to twist right to put the screw in, that's a right-handed screw. And if you have to twist left to put the screw in, that's a left-handed screw. And now, if you imagine spinning the screwdriver like in water, if you spin it one way, the water is going to go away from the screw. And if you spin it backwards, the water is going to come towards the screw. And this is kind of the idea behind runs and tumbles, is that the tails themselves are coiled like a screw. So when they spin in one direction, it causes the bacteria to go away from where they're spinning. And if it spins in the opposite direction, it sort of gets caught and the water is now hitting the bacteria, and it sort of it becomes more difficult to move. And so you'll see here in this diagram that the tails are sort of caught together in this screw-like bundle. If one of them starts going against the grain, it causes the whole thing to explode, until the cells eventually have them all going the same direction again. So sort of like how a snake, it's sort of like how a snake moves. And like imagine if you like held a snake by its head and it kept trying to move, it would sort of like flail everywhere. Yeah, that's the right mental picture. That's a great question.
Awesome. Another question is, how do bacteria tell if they're moving into a nutrient gradient?
Right. So we're gonna get to how they actually make these sense, how they actually sense things in part two, because we need to understand the specifics of that to answer the second part of the activity, which is how do I smell if there's candles everywhere? So yeah, leading right up into part two. Maybe I'll take one more question if there is one.
A general question is, why is E. coli a favorite cell used in the lab?
Yes. So E. coli is a favorite because for the most part, they're harmless. I think a couple years ago, there was an E. coli outbreak in cabbage, and then before that, there was beef. They're a favorite because for the most part, they're harmless. They actually live inside you. So the E. coli you use in the lab was actually harvested from some guy's butt in like the 1800s. So like he was sick, and so like they took poop samples from him, and then like sampled the poop and grew it in broth, and then that cell you like take their brothers and sisters and pass them to all your friends, and that's the E. coli we're left with today. So it sounds gross, but it's not so gross anymore. It's been a long time since we got bacteria from poop, but we use E. coli a lot in the lab because we've known about it for so long that we can actually manipulate it genetically. So it's really easy to make mutant, like weird E. coli versions. So there's a lot of reasons why it's super good. It's super good because we know a lot about it, because it's super old. They're really easy to get. They're everywhere. They're harmless, and we can make crazy mutant bacteria. So yes, great question. All right, I'm going to move on now. So somebody asked, how exactly do they tell the difference between, how do they sense when food is increasing or decreasing? And to answer that question, and to answer the question of what do I do when there's a lot of food around already, we need to know a bit of the specific details about how the sense of smell in bacteria works. And so turns out it's pretty similar to you and me. So when you and I smell, it's because in the like back of your nose, I actually don't know anything about neuroscience or like smell in humans. So just bear with me. But so like when you smell something, the general idea is that there's receptors in your nose, which are just molecules that stick to food. And when they stick to food, they tell your brain that there's food. And in bacteria, that's basically the same. You have these molecules that look like this. They're pretty cool. And what they do is they stick to food. So if they're stuck to food, they tell the cell to run. And how they tell the cell to run is actually a little bit complicated and not completely understood, but they tell the cell to run if they're stuck to food. And if they become unstuck from food, they tell the cell to tumble. So using these receptors is how the cell learns whether or not it's increasing or decreasing food. And I don't expect you to learn this in particular, but I just want you to see that these are actually really, really beautiful. So what this is a top-down view of the receptors. So here we're looking at the side. Here I'm looking at the top-down. And it turns out that they're arranged in these really beautiful patterns. And you can actually see these patterns if you have a really high-powered microscope. So if you have a really high-powered microscope and you zoom in really close, you can see these patterns on the surfaces of cells. So I think this is just something really beautiful. So it's just more reasons to study bacteria. They're so fun. But anyways, sort of related to this idea of run and tumble, because you can switch between saying to run and saying to tumble, these receptors sort of act like light switches. So they either turn on or off. They turn off when they're stuck to food, and they turn on when they're unstuck to food. And as I add more food, more of these receptors turn from on to off. So here where the food is really high, there's going to be mostly off receptors. And here where there's no food, they're going to be mostly on. But this becomes a little bit more difficult if I add food everywhere in the environment. So if there's candles everywhere, now the amount of food is very large. So even if I'm here, I'm at the bottom of the gradient, but the sum total of the food puts me at the bottom of the onness or activity. And even if I move up, that doesn't change the number of on or off receptors, no matter where I am in the gradient. So this is a problem. And to sort of understand how this problem works, I think it's useful to think about what happens when you wake up in the morning. So when you wake up in the morning, you're sleeping, and then you open your eyes, and then there's like intense pain. It's horrible. I hate waking up because everything seems super bright. And so what it is that your vision is saturated. So when you're asleep, you're in the dark. And so your eyes are expecting the light you see to be pretty dim. But when you open your eyes, all of a sudden there's light in the room and everything seems super bright. But as you all know, eventually your eyes adjust and things aren't, that picture's hurting your eyes. Sorry for hurting your eyes. I didn't take into account. But anyway, so like eventually your eyes adjust. And so this is the basic idea of what bacteria also do, which is that if they come into an area that has a lot of food that they aren't used to, eventually it's like their eyes are blinded, but they're able to adjust. And this adjusting is called adaptation. And cells have this really neat computer built into their genetics that allows them to do this adjusting. So to see what this adjusting looks like, this arrow is going to point towards the on-off curve that we're on at a certain amount of background food. So if there's no food in the background, we're going to be at this very leftmost curve so that we can tell the difference between increases and decreases of food above and below this level. Oh, whoops, wrong direction. But if now we have background food, so we've put like 1 or 2 candles in the room, now all of a sudden we push the curve to the right a bit. So now we can tell increases and decreases in food from this background level. And then if we add more and more food, we keep pushing the curve to the right. And so I hope you can see that this is how the cell solves problem 2. So as I add more and more candles to the environment, the cells adapt, their eyes adjust, or I guess in this case, their noses adjust. And it allows them to basically ignore the smell of the candle. Or in cell terms, it allows them to ignore the smell of the background signals. And so by doing this, by ignoring it, you take this signal that looks like this, you got a big background and some gradient on top of it, and you convert it to something like this, which we all established you're able to track using the run and tumble motion. So yeah, I guess I'll pause here for questions again. I have just a couple more slides at the end, but this is the most complicated part, I think, of this. So if anyone has any questions about sort of how this works, or if anything was unclear, let me know.
One of the questions is, why don't bacteria just keep going in the same direction when they sense that food is increasing?
Yes, so that's sort of a complexity that I've swept under the rug. So, but that's a really interesting question. That's something that I think about a lot. So if it senses, you're right, like if you're going up the gradient, you could be going at an angle. So the best way to go would be go straight, right up the middle, but you could be going at an angle, but the food would still be increasing. So you would still think that you want to keep going in the same direction. It turns out that bacteria over like millennia have learned that that isn't a good strategy because there could be a better direction to go. So pre-programmed into that system that tells them to run or tumble is an element of randomness. So this actually ties in perfectly to what we're going to talk about next. But in this system, there's an element of randomness, which is even if the food is increasing, sometimes at random, I'll switch direction, just in case that there's a better direction I could be going in. So yeah, cells actually use randomness a lot. Yeah. Any other questions? Or?
Sure. Another question is just, do bacteria use their smell to direct themselves?
Yes, definitely. But actually, turns out some bacteria don't only just smell, some actually see also. So here what we talked about is how the receptors basically act like smell. It tells them where food is. But some bacteria also have receptors that detect light, and those are super cool. So like echolocation with smell. Yeah, it is like echolocation with smell. You're seeing where the density, with echolocation, you're measuring the density of sound, and here you're measuring the density of food. But they can also do this with light. So sometimes if cells sense blue light, they'll swim towards it, and as they swim towards it, have you ever heard like an ambulance go by, and it seems like the pitch of the sound changes? That's because the sounds coming out of the ambulance get denser as it moves towards you. And that's the same idea behind swimming towards light, is as you swim towards blue light, the color of the light shifts very slightly, like ambulance sound. So yeah, there's a lot of cool things that bacteria do. Anyway, I only have 2 more slides. So in this one question, they asked, why would you not go in a straight line all the time? And it's because cells have an element of randomness. They can randomly switch their direction. Turns out that randomness applies to all things in all parts of the cell. So what they've done in this experiment is they've taken one of these E. coli and they've cloned it. They've made like 1 million copies of it, and they've given them all a red and a blue protein. So this is a molecule that glows, or not blue, red and a green. One glows green and one glows red. And then they've looked at these cells, and some of them are green and some of them are red. These cells are all identical. They're identical twins, but they're not all the same color. And this is super interesting because it means that even if you're genetically identical, there's going to be subtle differences between you and your siblings. And it turns out that these subtle differences, which we call diversity, which means that there's a lot of different colors in the population. There's a lot of different, in this case, swimming speeds in the population. This diversity actually allows populations to swim faster than they could if they were all the same. So what our lab actually did in this experiment is these red cells swim really fast and these blue cells swim really slow. And we've mixed them together. And you would think that the fast cells would pull away from the slow cells, but it turns out they actually swim as a group. And they swim faster than a single cell could on its own. So something really cool to look forward to in this field of microbiology, in this field of bacterial smell, is to understand how noise and randomness and diversity actually contribute to function. And that's the last idea I wanted to leave you with today. I guess the things we learned about, aside from bacteria being cool, is that run and tumble motion, this strategy bacteria use to find food, is very different from how you and I would solve problems, but it solves the same problem of locating the sources of smells. Adaptation is a thing that bacteria use just like you or I do, to adjust to background smells and adjust to brightness. And the last thing, and this is actually what our lab studies, is how diversity and cell-to-cell variability makes populations good at solving these kinds of problems. And you can prove it with math. So that's all I've got for you guys today. Anyway, happy to take any last questions if there are some.
Awesome. Thanks, Jeremy, for a great talk. There's lots of really good questions.
Great.
So one question is, can bacteria be airborne?
Oh, definitely. So this is my favorite. I don't know if this is true either, but I'm like 90% sure it's true. So bacteria are in the air everywhere, but turns out that snowflakes. So there was this experiment they did like 10 years ago or something, where they melted down individual snowflakes and then tried to culture them in like petri dishes. And at the center of these snowflakes was bacteria. So some people think that snowflakes are nucleated or like started out by a bacteria swimming in the air. So I don't know what happened to that line of research, but that was a super fun fact that I learned when I was in high school. That bacteria are in snowflakes.
Cool. Another question is about bacteria and their senses. So what senses do bacteria have? Is it anything like touch or hearing?
Yeah, so touch, I don't think they have hearing, but there was a paper so there was a group that was researching whether or not they can hear Wi-Fi. So there was a paper where they put some bacteria next to like a Wi-Fi port and some bacteria not next to a Wi-Fi port. And it turns out that like the cells near the Wi-Fi looked different than the cells not near the Wi-Fi. So I wouldn't worry about Wi-Fi at all, but like bacteria might be able to like hear, not really hear, but like sense those frequencies. As for touch, touch is actually related to flagella. So the flagella E. coli used to swim. E. coli doesn't do this, I don't think, but some other bacteria called Pseudomonas, they can actually use their flagella to sense whether or not they're near surfaces. So what they want to do is swim and land on a surface and then, start a family. And so they actually use their flagella to determine whether or not they're near a surface. So if the flagella is like hitting on the surface, they'll decide to attach there. So that's almost like a sense of touch. We've talked about smell. Some bacteria can sense light. Yeah, I don't know what the rest of the senses are, but no sixth sense. I don't think they have any sort of weird ESP or anything. But
a follow-up to that would be, how do bacteria react to spicy stuff?
Spicy stuff. I mean, first of all, what does it mean to be spicy? I think that's the question, right? It's like, what does it mean to be spicy? So in us, once again, I don't know anything about human beings, but in humans, there are receptors that like tell your tongue that something is spicy. I don't know. There's a lab at Yale that actually studies, or the boss of that lab at one point studied spicy things, or knew some of the studied spicy things. I don't really know much about spice, but like, spiciness is a sensation that like to you think something is spicy, but the thing itself like may or may not be spicy, which is also why like different people have different spice tolerances. So whether or not bacteria can sense whether something is spicy, I don't think they really know. I think they just know whether or not things kill them, and they don't have time to worry about whether or not they taste good.
Awesome. Another question is, how do bacteria know where their partners are so that they can coordinate? And a follow-up to that one is, what happens when bacteria bump into each other?
Mm-hmm. So this is very leading, and I'm not going to say anything more on that. But anyway, so bacteria don't have by partners, I think they meant like romantic partners, but maybe not. But anyways, they don't have romantic partners. They actually reproduce asexually. So they reproduce on their own. They just sort of divide in two, and then you have two bacteria, sort of like worms. Like if you cut a worm in half, it'll regrow into two worms. And bacteria do the same. They just like divide into two of themselves, but they do communicate with each other. And there's a word for that. It's called quorum sensing. And so the way that works is that bacteria always secrete these molecules that say, hey, I'm here. And then if enough bacteria are in a group together, there's a lot of this molecule that says, hey, I'm here. And it makes them change their behavior. So one of the bad guys we talked about, Vibrio cholera, that's what it does, is that each cell is always sending out this signal that says, hey, I'm here. And when there's enough of them, the hey, I'm here's tell the cells to start forming what's called a biofilm. So if you if you get like films on your teeth, that's like, or plaque, that's like a form of biofilm. And what it is like a big colony of bacteria that live inside you. So bacteria communicating is actually really bad for human beings. But sometimes there are Vibrio species that when they communicate do really cool things. So there's a relative of cholera called Vibrio harvii that when there's a lot of them, they actually glow blue. And so if anyone's interested, I'll type it in the chat, Vibrio harvii. I think it's this. I'm not actually sure how to spell, but if you put that in, you'll find it. And so if there's a lot of them, they'll glow blue. And turns out they live in the stomachs of these squids. And so these squids keep them in their stomachs to hide their shadows. So they glow blue so that at night, when the moon is shining on the squid will form a shadow, and predators will use the shadow to eat the squid. But if they get these bacteria in their stomach like a spotlight, it blanks out the shadow so that they can't be eaten. And so that's super cool. Yeah, somebody said bioluminescence. That's exactly it. This is an example of bioluminescence. Bioluminescence in Vibrio comes from them actually talking to each other. So yeah, that's really cool.
Awesome. Well, thank you so much, Jeremy, for a great talk.